Greetings, this is NFNF2552, Cosmeceuticals. This time, I would like to introduce the regulation of cosmetics contain nanoparticles by FDA. But first, what is nanoparticles? Nanoparticles are particles between 1 and 100 nanometers in size. Nanoparticles is small object that behaves as a whole unit. IUPAC define nanoparticles as particle of any shape with dimensions in the 1 × 10⁻⁹ meter and 1 × 10⁻⁷ meter range. Nanoparticles differ in chemical, physical, and biological properties from those of their larger counterparts. For example, their magnetic properties alter electrical or optical activity. This may affect the performance, quality, safety, or effectiveness of the nanoparticles contained in the cosmetic products. As we know that nanoparticles are so small in size, so the smaller the particle is, the increased surface area will be to the particles. Increased surface area to volume ratio, morphology, surface features, and charge can affect the distribution of materials in the body. So, proper identification of the chemical composition is needed. This including the impurities, structure, configuration of the nanomaterials, and the toxicology testing and stability testing are needed to be carried out. Another major part of the guidance is toxicology consideration. Both short-term and long-term toxicology of nanomaterials used in the cosmetic products should be addressed. This, including the possibility of ingredient-ingredient interaction or ingredient packaging interaction. This is very important to know that whether the nanomaterials will interact with other ingredients or the packaging of the cosmeceutical product. Traditional toxicity test method is used, but it depends on the character, this including the solubility, agglomeration, and aggregation of particles of the nanomaterial used in the cosmetic products. Some traditional in vivo test method suitable only for soluble nanomaterials, while some traditional in vitro and in vivo test methods needs to be adjusted for testings insoluble or partially soluble nanomaterials. The routes of exposure should be considered in those toxicology testing to ensure the safetiness of the cosmetic products. As we know that, although most cosmetic products are applied directly to skin, but some of the cosmetic products may come in contact with mucous membrane. Let me give you an example, such as eyeliner and mascara. Those eyeliners and mascara may smudge and causes the ingredients to in contact with the mucous membrane underneath our eyelids. Other than that, some may be applied by spray, such as deodorants and antiperspirants. There is a possibility to inhale the ingredients. Some products are applied at the parts where there is the possibility of oral exposure. This product includes lipstick and lip balm. So, those to the primary exposure organs and secondary exposure organs should be considered while toxicology testing is carried out. Now, this is the last part of the toxicology consideration, which is the toxicity testing. Those testing should include testing for acute toxicity, skin irritation, ocular irritation, dermal photoirritation, skin sensitization, mutagenicity, genotoxicity, repeated dose, which are carried out 21 to 28 days, toxicity, and subchronic toxicity, which carried out 19 days observation. Lastly is the phototoxicity testing. Thank you for watching. Don't forget to subscribe. Oh, just kidding. Have a nice day and goodbye.